DNA sequencing. DNA sequencing is the process of determining the exact order of nucleotide within DNA molecule. This method is used to determine the order of the 4 bases adenine, guanine, cytosine, and thymine in a strand of DNA. The advantage of rapid DNA sequencing method has greatly accelerated the biological and medical research. 4 nitrogenous base of DNA. Here cytosine, thymine, uracil, adenine, guanine, and this is structure of DNA. DNA sequencing has been feasible because of the following developments: available of restriction endonucleases, development of highly sensitive gel electrophoresis technique which can separate DNA fragment differing by only one nucleotide, availability of large quantities of individual DNA fragments due to development of gene cloning and PCR technique, development of reliable, easy, and rapid DNA technique. There are 2 basic techniques of DNA sequencing: chemical sequencing and enzymatic sequencing. Chemical sequencing explored by Max and Gilbert. Enzymatic sequencing explored by Sanger. This is the Max Gilbert procedure, chemical method. Alan Maxam, and Walter Gilbert published the DNA sequencing method in 1977. Based on chemical modification of DNA and subsequent cleavage at specific bases, this method allows purified sample of double-stranded DNA to be used without further cloning. Maxam Gilbert sequencing requires radioactive labeling at 5 end and 3 end of the DNA, followed by purification of DNA fragment to be sequenced. And this is the step: radioactive labeling of one end, cut the DNA fragment with specific restriction enzyme, denature the double-stranded DNA to single-stranded DNA by increasing temperature, cleave the DNA strand at specific position using chemical reaction. For example, we can use one of 2 chemical followed by addition of piperidine, dimethyl sulfate, selectively attract purine A, while hybridizing selective attack pyrimidine C and B. This is called the modification step. Chemical treatment generate breaks at 4 nucleotide bases in the 4 reaction mixture. And this is the technique in full detail. Double-stranded DNA, label the end with double-stranded DNA molecule, cut the end, label DNA strand with restriction enzyme, discard the short piece of DNA, denature the long double-stranded DNA, expose the 4 samples to different chemical reaction proceed long enough to produce an average of 1 break per strand. The random breaks generate in the labeled fragment representing all position of each indicated bases. Reagent mixture, reagent G, it breaks the DNA chain after guanine G base. Reagent AG, it breaks the DNA chain after adenine A. Reagent C, it breaks the DNA chain after cytosine C. Reagent CT, it breaks the DNA chain after cytosine and guanine. And these DNA modification steps destroy the base G, destroy base EG, destroy base TC, destroy base C. The concentration of the modification, the modifying chemical is controlled to produce on an average 1 modification per DNA molecule. Thus, a series of labeled fragments is generated starting from the radio-labeled end to the first cut. As a result, we have several different sized DNA strands in 4 reaction tubes. Fragments are subjected to electrophoresis in high-resolution acrylamide gel for size-based separation. To visualize the fragment, the gel is exposed to X-ray film for autoradiography which yield a series of dark bands which each corresponding to radio-labeled DNA fragment from which the nucleotide sequence may be inferred in gel. The fragments are ordered by size, and thus we can deduce the sequence of the DNA molecule, DNA sequence by Max Wilber technique. DNA sequencing evaluation, reading the gel. The gel is read from bottom to top. The gel has nucleotide sequence different by only one nucleotide. The larger the fragment, the more it's slowed down by the gel. Each band on the gel identifies the specific nucleotide. Enzymatic procedure, chain termination method. The enzymatic method is called a Sanger method. Sanger sequencing method was developed by Frederick Sanger and his colleague in 1977. The development of this technique won Sanger the Nobel Prize in Chemistry in 1980. From 1980 to the mid-2000s, Sanger sequencing dominated the DNA sequencing platform, bringing the successful completion of the DNA Genome Project in 2003. Although this technique has been replaced by next generation sequencing method, it's still used today for smaller scale projects. In order to perform the sequencing, one must first convert double-stranded DNA into single-stranded DNA. This can be done by denaturing the double-stranded DNA with sodium hydroxide. The Sanger reaction consists of the following components: single-stranded DNA fragment, template, a DNA strand, to sequencing one of the single-stranded, which was denatured using sodium hydroxide, all 4-deoxyribonucleic acid triphosphate A, DTATB, DGTB, DTTB, DCTB, or at least one of the 4-deoxyribonucleic triphosphate should be radioactive in each of the 4 reaction mixture tube in order to permit the auto-radiographic development of DNA bands after the gel electrophoresis. DNA polymerase, each incubation tube will also carry DNA polymerase enzyme sequenase in order to copy the DNA template by adding nucleotide to the primer in the synthesis process. Sequenase is engineered E. coli DNA polymerase 1, known as clinofragment, which is used to copy the DNA template. Sequenase is obtained by removing the first 3 to 3 amino acid of the polypeptide 5 to 3 using endonuclease enzyme. DNA primer, the enzyme sequenase need a primer to start end-to-end nucleotide synthesis. A primer should have the following characteristic. The primer should be either DNA restriction fragment or short DNA sequence complementary to the single-stranded DNA template. The primer should have free 3-OH group required to make 3 to 5 phosphodiester linkage between the nucleotide to be added to the primer. The primer should be actively labeled at 5M. For reaction mixture, in addition to radio-labeled primer and DNA polymerase, each incubation tube carrying a reaction mixture should have all the 4-deoxyribonucleotide DATB, DCTB, DGTB, DTTB, and a particular deoxyribonucleotide phosphate. The 4 reaction mixtures should differ from each other in having different deoxyribonucleotide phosphate analog. Example 2A will carry sequenase radio-labeled primer, all the 4-deoxyribonucleotide DATB, DCTB, DGTB, DTTB, and a particular deoxyribonucleotide phosphate DDATB in this case. Deoxynucleotide are chain elongated inhibitor of DNA polymerase used in the Sanger method for DNA sequencing. A deoxynucleotide DDNTB is artificial molecule that lacks hydroxyl group at both second and third carbon of the sugar moiety of DNA. In contrast, a regular deoxynucleotide triphosphate has the hydroxyl group in third carbon of the sugar. DNA chain component: the adenine, cytosine, guanine, thymine, here in 5,3 phosphoric acid and nucleotide. The main purpose of the 3-OH group is that it's used to form a phosphodiester bond between two nucleotides. This allows a DNA strand to elongate. During DNA replication, an incoming nucleotide triphosphate is linked by its 5-alpha phosphate group to the 3-hydroxyl group of the last nucleotide of the protein with DD and TB, normal nucleotide, and deoxy chain termination. Where there is 3-OH group, this reaction can take place, so elongation is terminated, but each new strand will stop randomly at position where D and TB is replaced by DDTB. The concentration of DD and TB should be 1% of the concentration of D and TB. The logic behind this ratio is that after DNA polymerase is added, the polymerization will take place and will terminate whenever a DD-ATB is incorporated into the growing cell. Thus, 4 sets of chain termination fragments corresponding A, C, T, G are produced in 4 reaction mixture. Procedure: the single-stranded DNA is mixed with primer and split into 4 reaction mixture. X reaction mixture contains DNA polymerase, 4 desoxyribonucleotide phosphates, and a replication terminator. DD and TB each reaction proceeds until a replication terminating nucleotide. Fragmentation and amplification, denaturation, attached to primer. 4 different reaction vials are taken, each with 4 strands of D and TB. After the occurrence of DNA synthesis, each reaction vial will have a unique set of single-stranded DNA molecules of varying lengths. Find the nucleotide sequence using gel electrophoresis. Enzymatic procedure, DNA sequencing. This is the steps. Autoradiography, in order to identify various terminating chain of DNA, the DNA strand, the DD and TBs, would have to be radioactively or fluorescently labeled beforehand. The DNA strands are then separated using gel electrophoresis, and the gel is read from top to bottom. Smaller strands migrate to the bottom, while large strands stay up top. The chain of termination method, not as toxic and less radioactivity than Max and Gilbert method, easier to automate. Lori and Hood and co-worker used fluorescently labeled DD and TBs and primer for the first high throughput of DNA sequencing machine. This lowered the cost from 100 million to 10,000 USD in 2011. Highly accurate long sequencing rate of about 700 base pair. Easier to get started. The kits that are commercially available contain reagents necessary for sequencing, pre-allocated and ready to use. Automated DNA sequencing, modification of chain termination method. Automatic DNA sequencing is based on Sanger method of DNA sequencing. It's make use of automatic sequencer. Steps labeling the product of polymerase chain reaction. In place of radioactive labeling, fluorescent labels are used. The fluorescent labels are attached to the 4-deoxyribonucleotide if different. Fluorochrome is attached to each of the 4 DD and T. All of them may be used in the same reaction. In a single track system, the reaction products are run in a single lane of gel or capillary. The reaction products are subjected to polyacrylamide gel electrophoresis under the nature condition. The band produced in polyacrylamide gel are identified with the help of fluorescent detector, which identify the fluorescent signal emitted by each band. Automated DNA sequencing. The detector shortly labeled strand. Automated DNA sequence. The fluorochrome are excited by laser beam, and the resulting fluorescent signal is sensed by photovoltaic cell. The resulting data are fed into a computer, which converts this signal into the base sequence of DNA molecule. The sequence information can be printed out or stored in data storage device for future use. Automated DNA sequencing has the following advantage. Radioactivity is not used, gel processing after electrophoresis and autoradiography is not needed. The tedious manual reading of the gel is not required as the data are processed by computer. The sequence data is directly fed and stored in the computer. The separation of the same reaction product can be repeated to check the result in case of doubts. The method is extremely